Pop up. Oh, okay. Oh, we know that's running. Great. What about iPhone? Okay, great. Awesome, bro. All right, big dogs. So, today was an excellent day, and you know what I realized? I was thinking about, oh, hello, my fellow risers, and welcome to the Pop Bro. So you know what I was thinking about as I was thinking about what I'm gonna say at the beginning of this stream. I want to tell you, it's been a lovely day. Especially because I was really worried about something, about Sarah's dog. Especially, I was worried about it being my fault. I mean, I was worried about the dog, and I was especially worried. Oh, I'm not going to compare which I was more worried about. I wouldn't know how. But, anyway, so what a relief that he is going to be okay. And, um, even if he did eat a chicken bone out of my trash, um, that is not the issue. So that's really exciting. Um, on top of that, well, it's a good day for another, but a goofy reason. A goofy reason. And then also cool because I'll see my sister tomorrow. She's coming into town. So anyway, we're feeling good. And also my desk is clear and I really like the setup. Um, we'll clear that stuff. But I actually love this view from the back. I think it's pretty cool. So, anyway, let's read a bit. We were reading Ramachandran. So we have to, there he is. The Ram Ram. So that's good. And then we need just to zoom in on that. Pop, pop. Or did we go with another image? No, this was the one, right? And then we need this open. Great. Zoom in on that a bit. Great. And we are all set to read more of the Ram Ram. So, Um, you know what? I'm actually super tired, so I don't think I'm gonna explain a lot right now, but maybe I'll wake up and explain some as I read. Um, because remember, he's talking about am, a, apo, apotemnophilia, which is the love of like someone who really wants to amputate something. And it's not because they don't think it's theirs, which I had thought as I was reading it. Um, They know it's theirs. So I still have the line, I think. Very slight, you can't see it. But anyway, so I have this patient that drew this like squiggly line. It was like exactly the same when he did it months later. So it's clearly something he's really experiencing. Um, anyway, so we'll start at near the top of page 75. Here's a bizarre psychological syndrome. The person wants his arm removed. And he's already been talking about this. Here's a bizarre psychological syndrome. The person wants his arm removed. Discard the Freudian idea that he wants a big giant stump or that he wants attention and things like that. And you come up with a pretty precise circuit diagram of what's going on. We tested this because it is not enough to come up with a theory. How do we test it? It turns out that if I poke anybody with a needle, that pain sensation. Hold on, I just want to see how well that's working. Oh, that was just my computer that made that noise. Okay. Um. Okay. How do we test it? It turns out that if I poke anybody with a needle, that pain sensation goes to the sensory pain region in the brain, probably in the thalamus and cortex, and then it goes to the amygdala. The amygdala alerts you to the pain, and you say, ow, right? And then it goes down to the anterior cingulate that feels the agony of the pain. There's a cascade of events. There's a sensation of pain, and then the agony of pain. And then messages go down the autonomic nervous system and make you start sweating, preparing for action, fleeing, fighting, or whatever the required action is. So if you poke somebody with a needle, this whole cascade of events is set in motion. You can measure the skin resistance, which measures the sweating. You start sweating when poked. Now, when I poked him with a little pencil above the line, he wanted his arm to be amputated. Nothing happened. So the part that he wanted amputated, when that got poked by Ramachandran here, nothing happened. Just a little gentle pencil prick. It's not a painful stimulus. Not much happened. There's no galvanic skin response. There's no arousal. But if you touch him below the line where he wants the amputation, there's a huge, big galvanic skin response. You're right. We should make sure, because that was weird that he wants like this part of his arm amputated. Did we misread that? Because I'm actually a little bit confused because I don't understand. It's not a painful stimulus. So why would there be arousal? And then you touch him below the line where he wants the amputation, as opposed to above the line where he wanted his arm to be amputated. So you touch him below the line where he wants the amputation, and all of a sudden you have an autonomic response. There's a huge, big galvanic skin response. You can actually measure the aversions physiologically, not just rely on the subject to report. So which part does he want amputated? He wants his left arm amputated above the elbow. And he freaks out when you touch below the elbow. But you're telling me it's not a painful stimulus. I'm a little bit confused. But maybe we'll understand in a moment. So anyway, you can actually measure the aversion physiologically, not just rely on the subject to report. There's no way you can fake the galvanic skin response. It's the basis of all lie detector tests. Then, of course, we went straight to the brain and said, "Let's map it out." And as I said, no, I have to find out. I have to find out. Damn it. No biggie, bro. There's always an update, isn't there? Oh, let's share my screen. Oops. Mac OS screen. And we'll put that above the Ozbot. Oh no, not that screen. This screen. Put that above the OBS bot. Here we go. Alright. Apotemnophilia. Why am I not understanding? I'm confused, bro. Oh my God. I get it. He's saying cut right above the elbow and get rid of everything below it. I got you. But yeah, amputation, that's the actual cut. For some reason I was thinking like he wants the material above his elbow amputated. All right, I think I get it now. No, bro. No, this is I'm just an idiot because of course it's a stump. So it's just he wants it cut right above the elbow. All right, I'm just an idiot, and that's okay. We forgive me. All right, great, great. Okay, I'm all clear now. So before we look to make sure we understand, so we do a gentle prick, and it's not a painful stimulus. So we do that above the line, and there's no response. But then when we just even touch him here, which is what he wants cut off, which is what I finally understand, all of a sudden there's a big response, big distress. So there are parts of him that know that this is his, but according to ChatGPT, there is some alienation. There are parts of him that don't feel like this is his, and that's why they freak out when he feels something. Okay, we are all caught up. Okay. Now, when I poked him with a little pencil above the line where he wanted his arm to be amputated, nothing happened. Just a little gentle pencil prick. It's not a painful stimulus. Not much happened. There's no galvanic skin response, there's no arousal. But if you touch him below the line where he wants the amputation, there's a huge, big galvanic skin response. You can actually measure the aversion physiologically, not just rely on the subject to report. There's no way you can fake the galvanic skin response. It's the basis of all lie detector tests. Then, of course, we went straight to the brain and said, let's map it out. As I said, we found S1 was normal. Okay, so this darker gray here is normal. The thing right next to the red, can you tell the difference? Oh, great. Look at that. That's great. Okay, so this, normal. I'm trying to touch the gray here. Yeah, that's normal. But the supplementary sensory cortex, which integrates the information in this superior parietal lobule, not normal. So if you go to S2, and as I said, we found S1 was normal. Oh, if you go to S2, that's normal. Hold on. I'm sorry. I just lied to you, apparently. I'm sorry. I'm sorry. So, parietal up, which is located within the lateral sulcus. So here we have our lateral sulcus. So, within the lateral sulcus and inferior to S1. So this is visual representation. Hold on, bro. All right, we're gonna look it up because I'm actually still a little bit confused, bro. Okay, we have S1, inferior parietal. Um, S2, and that's inside the lateral sulcus. Oh, so does S1 just sort of fold into S2? I did not know that. Okay. Ah, got you. Got you. Okay. So, so this is all the let me turn it the right way. Oh, it's gonna be confusing for me. Hold on. Okay, so we have S1, okay, and then as S1 comes in here, it goes into there because this is the operculum, whatever, and in here, in there, just before the insula, is S2 as shown here. Okay, and then the superior parietal lobule is here. And we're going to confirm that. But yeah, okay. So here we have S1. And then in there is S2. This is our lateral sulcus here. Um, yep. And then here, great, we have the superior parietal. Like here, basically. Or like there. Oh, this whole thing, I guess. This area here. Okay. Okay, so in S1 and in S2, no problem. But then the arm was not. So the arm was represented here and here. But then here, where information from here is integrated, the arm was no longer represented. I think we've got it now. Okay. So, and as I said, we found S1 was normal. If you go to S2, that's normal. If you go to the superior parietal lobule, where the body image is constructed, to some extent the inferior parietal lobule, right parietal, let's say, where the body image is, there is no arm representation in that center. Okay, that's what we found. If you touch the arm, there's no activity there. So they do some imaging. You talked about all the different imaging things. You do some imaging and maybe you see, oh, I shouldn't be pointing like that. You do some imaging and you see the arm's like up here, right? Yeah. You see something light up here from the arm. Maybe you see something in S2 light up. And then no activity here and here all of a sudden. So if you touch the arm, there's no activity there. But if you touch above the line of amp, oh, okay. But if you touch above the line of amputation, it's normal. The activity is completely normal. If you touch above the line of amputation or touch the normal arm, which is his right arm, and I know I keep demonstrating with my right arm, but anyway, which is his right arm, because I should have been demonstrating my left. The activity is completely normal. So that region of the brain is abnormal, but we also speculate that in the regions of the brain in the frontal lobes and insula slash amygdala, to which that superior parietal lobule slash inferior parietal lobule projects, there could be an interruption of signals. So I guess there's a pathway from here to, and we had an image of the so here's the insula, and so there's a pathway from here, the superior parietal lobule, the inferior parietal lobule, and then here's the white matter pathway, bop, down to the insula, and and the amygdala, which is somewhere there. I'm gonna be honest with you, I do have to work on my anatomy. Anyway, so there's some kind of an interruption in the signals here. So okay, so there's perfectly fine activity here. But then oh, okay, so there's a problem here in the superior parietal lobule and maybe the inferior. And then they also suspect a problem with the pathway down to the insula. In either event, there's a physiological reason why this happens. This is giving you insight into how the normal brain constructs a body image. At this point, I should add a note of caution that unlike our work on phantoms and synesthesia, which has been confirmed on dozens of subjects, both by us and others, this work on apotemnophilia is very preliminary. We need more subjects. And that's not a legal disclaimer, it's the truth. Let's wait and see. Then the question is, can you treat the guy? And we're working on that. We don't know how. In the case of phantom limb, there are ways of treating phantom pain. In the case of phantom limb, there are ways of treating phantom pain. And well, that's the mirrors, you know that. Anyway, we don't deliberately go after these odd phenomena. Somebody phones me and says, I have this curious phenomenon, Dr. Ramachandran. Can you solve the problem? 90% of the time, we can't. But every now and then we discover something amazing, as I said. So what we do is we look at the patient, and the first question is, why does this patient have this syndrome? Why this peculiar behavior? What's going on in the brain? Can you explain it? First of all, show that he's not crazy. Show that it's a real, authentic syndrome. There are bogus syndromes, and I will talk about that if you like. They're not real. But given that it's a genuine syndrome, and you prove that it's authentic, which is often very difficult to do, then, having done that, you say, what are the precise brain mechanisms that give rise to these curious symptoms? So the first two questions are, is it real? And if so, what causes it? The third question is, who cares? Why does it matter? What does it matter? Each of these three questions needs to be answered if you want to make progress, if you want to draw people's attention to the phenomenon that you're studying. Is it real? What causes it? And what the fuck does it matter? So let's take synesthesia. First thing we did was to show that these people are not crazy. They're really seeing colors when they see numbers. They're not just making it up. The second thing we did was to ask, what are the brain mechanisms? What's going on in the brain? Ed Hubbard, Boynton, and I have shown that when they see non-colored numbers, the color area in the brain lights up. So what? Why should I care? We've shown that this quirky phenomenon has broad implications for understanding creativity and metaphor. Well, we haven't actually shown it, but we speculated on that possibility. So in each case, what we do is we rescue this phenomenon, this anomaly, from being brushed under the carpet. Find out what the mechanism is in the brain and talk about its deeper implications for all of us, for normal behavior. Now, we seek odd syndromes to try to explain the symptoms in neural terms and hopefully shed light on aspects of human nature that have remained ineffable for the longest time. But sometimes you come across a syndrome where you cannot quite know for sure if this is a legit syndrome or not, even though you can find it in the Bible of Clinical Psychologists called DSM, Diagnostic and Statistical Manual, which is the official book for clinicians. If they can label you, give your syndrome a name, they can charge an insurance company. So there has been a tendency to multiply syndromes. There's one called, by the way, chronic underachievement syndrome, which in my day used to be called stupidity. It actually has a name and it's officially recognized. Then there's a syndrome called Déclermont syndrome. Déclermont syndrome refers to, believe it or not, a young woman developing an obsession with a much older, famous, eminent, rich guy and develops the delusion that this guy is madly in love with her, but is in denial about it. This is actually found in a textbook of psychiatry, and I think it's complete nonsense. Ironically, there's no name for the converse of the syndrome, where an aging male develops a delusion that this young hottie is madly in love with him, but is in denial about it. Surely it's much more common, and yet it doesn't have a name, right? You have to have a nose for anomalies, nose for the right kinds of syndromes to pursue. I'll give you examples, and I think two will suffice. Let's return to synesthesia, which I've been excited about studying for the last 3 or 4 years. It's not really a neurological syndrome, but it is an anomaly of sorts. Francis Galton described it in the 19th century, the great Victorian polymath who was a first cousin of Charles Darwin. Galton noticed that some people in the population were otherwise quite normal, except they had one quirk. Every time they saw a number, the number would evoke a specific color. So 5 would be tinged red, 6 would be green, 7 would be indigo, 8 would be blue, and 9 would be orange or something like that. Also, sometimes in the same subjects or in other people, each tone would evoke a color. So F sharp would be blue, C sharp would be green, so on and so forth. Galton also noticed that this runs in families and may have a genetic basis, and published this. I think it was in Nature in the 19th century. Since then, there have been dozens and dozens of case reports of people experiencing this, but it was regarded as a curiosity mainly, and is also thought to be very rare, estimates ranging from 1 in 1,000 to 1 in 10,000. I became very intrigued by this phenomenon, and I said, why would somebody see 5 as red? Now, as I said, it's been known for a long time, for more than 100 years, and people ignored it because it didn't fit the big framework of science. What do you make of someone who says F sharp is blue and C sharp is green? It doesn't make any sense because we didn't understand that we were brains, so. Or 5 is green, or 5 is red, and I tend to get intrigued by these phenomena. I said, well, what's going on in their brain? The first thing we wanted to show was that this was a legitimate phenomenon, that these people are not making it up. There are several theories of synesthesia, and I say that we didn't know that we are brains, which is not necessarily true, but I mean that the people who were like, oh, it doesn't make any sense, maybe they just thought we were like perfectly wired brains. But once you know we're brains and you know the quantity of cells and pathways and the possibilities, it's like, well, clearly just, there can be a failure, an interruption of signal, some kind of a failure of connections, and that's going to make a big difference and weird shit can happen. Right, Uncle?
All right.
One theory is that they are crazy. Maybe, but let's set that aside for a minute. One of the things we learn in medicine is that when a patient is trying to tell you something and you think he's crazy, it often means that you're not smart enough to figure it out. Sometimes he's crazy, but usually it means you're not smart enough to figure it out, so look carefully, talk to the patient. In the case of synesthesia, another odd aspect of it is that it's much more common among artists, poets, novelists, and other creative people. In fact, 7 or 8 times more common. This is controversial, but the strong evidence is that this is true. Now, why would that be the case? I mean, one of my students has shown this to be the case. Ed Hubbard. Why would this happen? There are several little mini mysteries here about synesthesia. Why would it run in families? Why would they say numbers are colors or tones are colors for that matter? Why would it be more common in artists, poets, and novelists, so on and so forth? So it's a medical mystery worthy of Sherlock Holmes, waiting to be solved. The first thing we want to show is that these people are not crazy. By the way, another common theory is that they are on drugs like LSD, acid junkies, or potheads. Sure enough, it's more common among people who are high on acid, but that makes it even more intriguing. Why would some drugs produce this merging of the senses, this peculiar phenomenon of numbers evoking colors? Another theory is that they're being metaphorical, as when you and I say it is the east and Juliet is the sun, or we just say this is a loud tie. The tie isn't loud, doesn't make any sound. Why do you say it's a loud tie? Or cheese. Cheddar cheese is sharp. Now, sharp is a tactile adjective, a sharp nail or something. Why do you use a tactile adjective to describe a taste, a gustatory sensation? You say, well, it's a metaphor. That's circular. Why do you want to use a tactile metaphor for a taste sensation? Explaining synesthesia as just a metaphor doesn't explain anything because it's trying to explain one mystery with another mystery, and that doesn't work in science. Another example of a metaphor would be it is the east and Juliet is the sun. You don't say Juliet is the sun. Does that mean she's a glowing ball of fire? No, you don't say that. You say she is warm like the sun, she is radiant like the sun, is nurturing like the sun, is a celestial body like the sun, a pun rather than a metaphor, is the center of the solar system, and so on. The brain forms the right links. Synesthesia, by the way, is a completely arbitrary link between five and red. It's not a metaphor in that sense. So I was uncomfortable with the idea, but I thought there might be something to it. But we'll come back to that later as we go along. About a decade ago, by the way, I proposed there may be unconscious synesthetic propensities in all of us, which has now been amply confirmed in many studies, including a recent one from Oxford. Another theory is that they're remembering childhood memories. Maybe they played with refrigerator magnets, and 5 was red, and 6 was blue, and 7 was green, and for some reason they're stuck with these memories. Well, this again begs the question of why you and I have played with magnets, but we don't have synesthesia, presumably. Most of us don't. We found, by the way, the phenomenon of synesthesia is quite common. You see it in 1 in 50 people. It's not 1 in 1,000 or 1 in 10,000. People often don't come out and say that they do because they're worried you might think they're crazy. So the childhood memories thing doesn't work because, as I said, why would it run in families? Another reason for not believing it, you would have to say the same magnets were being passed from generation to generation, and that doesn't make any sense. Metaphor? Maybe they are being metaphorical in some sense. Maybe it's related to metaphors. They're crazy? That's not a real argument. They're on drugs? No, that doesn't work either. Hold on a second. I just gotta check the cameras. Make sure Ankh is chillin'. Oops. You don't need to see the cameras though. Alright, great. Alright, great. Alright, drugs? They're on drugs? No, that doesn't work either. The first thing we wanted to show is they're not crazy. They aren't making this up. By the way, remember, this I think was spoken, which is why it sounds like this. They aren't making this up. We generated a computerized display made up of fives. Oh, bro, can I just finish this chapter? I love when you hang out. Oh no, I'm not gonna be able to finish this chapter. Oh, it's another 17 pages. We'll try to finish, but not before I let you out. So we generated a computerized display made up of fives, lots of scattered fives on the screen. And among those fives were scattered some twos. When you look at a 2 and a 5 is a mirror of a 2 in a sense, in terms of its shape. So you have a bunch of outline drawings of fives. Scattered among them are some twos forming a shape. The twos cluster to form a triangle or a square or a circle, like your Ishihara color test in traffic when you're going through a traffic school eye exam for color vision. It's similar to that. Oh, yeah, I didn't know what they were talking about first, but we know what they're talking about, right? Um, a normal person looking at it, the non-synesthete looking at this, says, "Oh, fives? You mean there are twos in here embedded? Let me see. Oh, there's a two there. There's a two. Okay. Oh, there's a two there. Oh, there's another one there." They take 20 or 30 seconds to find the hidden shape. A synesthete who sees five as red and two as green instantly sees a green circle or a green square or a hidden green shape pop out from the background. He's much faster in detecting the circle or the square than you and I are. If he's crazy, how come he's better at it than us? Secondly, if you ask him what he sees, he says, "I see a green triangle, I see a green square." Phenomenologically, perceptually, he literally sees the green square or the triangle or the rectangle. What this suggests is that it's a sensory experience, not a memory association, at least in some synesthetes. Jamie Ward has recently replicated our findings. Ishihara, just so everybody knows what I'm talking about, Ishihara synesthesia. So it's like this, you see, except not like this. But if you imagine that each of these green circles here is the number two, but except it's not in green, it's black. Imagine that all the brown is also black and all the brown circles are fives. Well, then you would, we should, okay, so like this, you see? So, um, and then for them, each number has a different color, right? Um, so they are immediately seeing like how these fours go whoop 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 whoop. I have to like find the fours, but they just see a line going. Um, Here it is. Here we go. So we're like, oh, here's a two. Oh, here's a two. Oh, here, okay, here, blah blah. But they've already seen them in. Here we go. So like we see this, but they see this. And then if it's a shape, here we go. Here we go. Okay, so now instead of the twos forming the shape, it's the fives. So we would see this as all black. And we would be like, oh, I see. There's a five. There's a five. Not this image, obviously. This image is what the synesthete sees, not what we would see. Um, so we're like, oh, there's a five. There's a five. Okay. Oh, I see. Oh, I guess it forms a triangle. But they look at this and they're like, yeah, I see an orange triangle. Okay, I think that's beyond fucking clear. So. It turns out there is a heterogeneity of synesthetes. There are some synesthetes whom we will call lower synesthetes, in whom the color is actually perceptually evoked and the numbers seem tinged with color: red, green, blue, yellow, chartreuse, or indigo. But there are also more conceptual synesthetes, where it does seem to be more like a memory association. We were focusing on the perceptual synesthetes, sensory synesthetes, the lower synesthetes, because they are easier to study scientifically. First, we've shown they're not crazy. It's a real phenomenon. Remember, I had 3 steps. First, to show it's real. Second, what are the brain mechanisms? Third, what are the broader implications? Why should I care? We've solved the first problem, which is to show that it's a genuine phenomenon. The second question is what causes it? Well, Ed Hubbard and I were looking at brain atlases, and we were struck by the fact that there's a structure called the fusiform gyrus in the brain buried inside the folds of the temporal lobes. This structure, the fusiform gyrus, it turns out, is where the color area of the brain is V4, which was discovered by Samir Zeki. Right next to it, almost touching it, is the number area of the brain. It represents the visual representations of numbers. The two areas are almost touching each other. We said, what's the likelihood that the most common type of synesthesia is the number color synesthesia, and the number region and color region are adjacent to each other in the brain? Oh, we said, what's the likelihood that the most common type of synesthesia is the number color synesthesia, and the number region and color region are adjacent to each other in the brain? This seems unlikely to be a coincidence. Then we said, maybe there's an accidental cross wiring between these two regions of the brain. How do you prove that? We did brain imaging. You take a normal person and do an fMRI or MEG and show them numbers. Only the number area in the fusiform gyrus will light up. Show colored numbers to a normal person, and V4, the color area, and the number area will both light up. If you show a synesthete a black and white number, both the number area and color area light up, thereby directly proving that there's a cross activation going on. Now, I should add that 3 out of 4 groups has shown it to now, I should add that 3 out of 4 groups has shown it to be the case. There's one group who claimed they don't see the activation. Research groups, I say. There's always uncertainty in brain imaging. Inherently, there is some noise, so it has not entirely been nailed down, but I think it's very likely that we are on the right track. Ramke Rolte in Amsterdam has studied this, and she has also shown that there is an actual increase in white matter, actual fibers connecting V4, color, and the number area within the fusiform gyrus. So that's about as good as it gets. Fusiform, I think fusiform is gonna be like, well, it's in the folds of the temporal gyrus. I think it's like here somewhere, isn't it? Maybe a little bit farther back. I mean, it should be pretty close to the occipital lobe. So maybe it's like here. Let's see. It's straight up inside. Oh, okay, okay, okay. So I can't actually show you on this. Okay. 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 So it's here. So then where's V4 exactly? V4 isn't all of that, is it? Ooh la la. Oh my God, it's so hot, bro. Okay, so the fusiform gyrus is all that stuff, and then you have shape. What is this?
Fusiform gyrus. Fusiform gyrus.
All right. Um.
Gyrus. Fusiform gyrus.
Okay. So we have shape, base, place. Great. And what about color and numbers so we can see?
A region in the inferior temporal cortex, fusiform base area that works for recognition of objects, faces, and facial expressions.
I am sweating, bro. Um. Uh-huh. V4. There it is. Okay, very good. You guys see V4 here? It's that little spot right there. Um, okay, here we go. Fusiform, V4, great. And then I guess number is just gonna be, you got such a hot flash, bro. Okay. Alright, very good. Okay. This is a visual pathway. So we have it. This is the thalamus, the LGN, to the occipital lobe, and then through the temporal lobe. So the I guess that's inferior temporal. Okay, very good. All right, I'm over that. Okay. Oh my God. I don't know how I just got so hot all of a sudden, but I'm okay now. Um. So Ramke Rolte in Amsterdam has studied this, and she has also shown that there is an actual increase in white matter, actual fibers connecting V4 color and the number area within the fusiform gyrus. So that's about as good as it gets. Now, why would this happen? Why do these people have this cross wiring? That's the next question. A clue first comes from observations made by Francis Galton and has been confirmed since then. It runs in families. It may have a genetic basis. So we said that if you take the infant brain, a fetal brain, there's a tremendous redundancy of connections. Everything is connected to everything. It's a crude approximation, but it's almost true. Then what happens is that there are pruning genes which prune away the excess connections between adjacent brain regions or even separated regions that were densely connected. This creates a characteristic modularity of the adult brain. Now, if something goes wrong, modularity, like different parts kind of doing their own thing, some kind of localization to parts or networks, whatever. Now, if something goes wrong with the pruning gene, if pruning fails to occur in adjacent brain regions, like the color and number areas remaining connected even in the adult, if the gene is selectively expressed in the fusiform gyrus through transcription factors, for example, if it's expressed in the fusiform gyrus, then you'll get a number/color synesthete. Every time your guy sees a number, because of the cross wiring, the color neurons are going to be activated. Every time he sees a number, he sees a color. In our early papers, we noted, I'm thirsty. I'm sorry. I'll be back. Try to knock this out in a half hour. Then we'll take Neo for a walk and turn in. Okay, so okay, pass out and mm, oh, he's about to talk about Juliet again. Juliet is the sun. I still don't really understand that. It is the east and Juliet is the sun. Hey, I mean, I don't remember that or remember anything from Romeo and Juliet. So, um, anyway, moving on. In our early papers, we noted that such cross activation could also be based on transmitters that cause disinhibition. Probably both things are going on. Voila, you explain number/color synesthesia. You started with a gene. The gene has not been cloned yet, but people are trying. You explain what's going on in the brain, why these people have these quirky visual experiences of seeing colored numbers. The last question, why should I care? The answer comes from the observation, the claim, that synesthesia is 7 or 8 times more common among artists, poets, and novelists. Artists like Kandinsky. Gosh, I know I probably never said that name, but I've heard it so many times. Artists like Kandinsky, for example. Why would this be the case? What do artists, poets, and novelists have in common? They're all very good at metaphor and analogy, seeing hidden links that most of us lesser mortals have difficulty in seeing. So when Shakespeare said, "It is the east and Juliet is the sun," as I have said before, you don't say Juliet is the sun, meaning that she's a glowing ball of fire. You make the right links. You say she's celestial, like the sun. You make any number of links. She's the center of my solar system, like the sun is the center of the solar system. She's radiant like the sun. She's warm like the sun. She's nurturing like the sun. Shakespeare was very good at picking these metaphors, which have multiple layers of metaphors and resonance. What has this got to do with synesthesia? What's going on in a metaphor? You're linking seemingly unrelated concepts and ideas, right? If the same synesthesia gene, instead of being expressed selectively in the fusiform gyrus and producing this quirky phenomenon of number color synesthesia, if it were to be expressed throughout the cortex, throughout the brain, it's going to create a higher propensity and higher opportunity to link seemingly unrelated ideas and concepts in far-flung brain regions. If we think of ideas and concepts as also located in specific brain regions, occupying specific brain regions, and if you have these long-range connections, then it permits greater opportunity for linking seemingly unrelated concepts. Hence the basis of creativity and metaphors. Hence the 8 times higher incidence of synesthesia among artists, poets, and novelists. In other words, what I'm getting at is that an evolutionary biologist could ask the question, what use is this gene? It's seen in 1 in 50 people. It's fairly common, not rare. It's fairly common, not rare. Why is it conserved in evolution? If there's a gene in evolution that's useless, it's completely useless to see 5 as red and 6 as green. It would have been eliminated from the gene pool eons ago, 10,000, 20,000 years ago. Clearly, this gene has been around and has been conserved. Now, why? Why is this gene still around if it's completely useless? Well, one possibility is that it confers some outliers in the population with the ability to link seemingly unrelated ideas, making them artistic, more creative. But when I give these talks, people often ask me why. If it's that good that that gene makes you artistic, creative, and metaphorical, why doesn't everybody have it? Well, it's a silly question because evolution takes time. And given another 20,000, 100,000, 50,000, all right, years, everybody will have this gene and will all be creative. But that's not the right answer. It may be a partial answer, but the real answer, I think, is that you don't want everyone being creative. We need engineers. You can see what we've done here, as with apotemnophilia, but even more so with synesthesia. It started with this peculiar phenomenon where people see number as color or tones as color. Then, from that, we said, what is it? Is it real? We showed that it was a real phenomenon using a number of perceptual psychological tests that can't be faked. From that, We went to the brain anatomy, doing brain imaging and showing what parts of the brain are involved. Then from that, we were able to say there's a genetic basis. So from gene to neuroanatomy to perceptual phenomenology. Finally, all the way to metaphor, Shakespeare, and poetry. All from starting with his peculiar quirk called synesthesia. This is what we do with every one of our syndromes. Sometimes we're partially successful. Sometimes we're fully successful in doing this. Given that our lab is well known for studying these odd quirks of human behavior and explaining what's going on in the brain and showing there are broader implications for understanding human nature, human consciousness, these things that everybody is curious about, when people have something quirky, they come and phone me up. Or if a physician stumbles on a new case which he finds he can't explain, he or she will often phone me. 9 out of 10 times I can't do anything about it, but every now and then I find what's going on in the brain and discover it's something very intriguing and possibly important. In the case of synesthesia, it was regarded mainly as a curiosity and an anomaly. People were just brushing it under the carpet and saying, what do you make of somebody who says red is 5? They're just making it up or they're crazy. That's why it's more common among artists, because we all know artists are a bit crazy anyway, and they all want to draw attention to themselves. There are all kinds of silly theories floating around. In fact, some synesthetes were diagnosed as psychotic and diagnosed as having schizophrenia. They were told they were hallucinating colors. They were prescribed psychotropic drugs for the schizophrenia. Then they came to realize that they had this perfectly normal phenomenon, not normal, but not pathological either, phenomenon called synesthesia. I think it's fair to say that we, and Jamie Ward, and Julia Simner, and a couple of other groups came and revived interest in this field, brought it into the mainstream. So now there are about 20 or 30 books on synesthesia. Not 20 or 30, maybe a dozen books on synesthesia. On just this one topic, though. When I started this nearly 10 years ago, nobody was interested in this topic. There was one book by a clinician, a neurologist named Chitovic. But he was a prophet talking in the wilderness. Nobody paid any attention. He didn't really do any definitive perceptual experiments on it. He just said, here's a phenomenon worthy of study. Here's a phenomenon worthy of studying. We were the first to actually start doing experiments on the phenomenon to show that it's authentic, show that it's perceptual, and then pin it down to brain anatomy. The reason I was attracted to it was because I'm curious about neurological syndromes given my background in clinical neurology, among other things. I began with being intrigued by phantom limbs. Patients would come into the clinic with an arm missing or a leg missing and continue to vividly feel the presence. Fuck. I was so excited about these. Finally a nice thick one that can stay on my wrist. Patients would come into the clinic with an arm missing or a leg missing and continue to vividly feel the presence of that missing arm or leg. And again, it has been known for about 100 years. And people thought of it as a curiosity, as a case of, hold on. I want to do this a little bit differently, actually. Nope, that's silly. Okay. And again, it has been known for about 100 years, and people thought of it as a curiosity, as a case study to be reported during grand rounds. Here is a patient with phantom limb. Nobody knew what to make of it, and certainly there was no interest in mainstream neuroscience in phantom limbs. What we found is quite intriguing. 2 or 3 things. One discovery goes back about 15 years. Let's say I'm the guy with the phantom limb. I've lost my left arm, and you're the physician. Fuck, that hurt. You come and touch the left side of my face. I start feeling the stroking sensation in my phantom thumb. I start feeling it in my thumb, bro. Even though I don't even have my whole fucking left arm. So I start feeling the stroking sensation in my phantom thumb when you're touching me here. Even though you're stroking my face, I feel it in my phantom. Is that because the hand and face are next to each other? Ah, fingers, thumb, eye, nose, face. Oh. Um, all right, whatever. If you touch this region, if you touch this region, it's my index finger. That's my pinky. If you touch this region, it's my index finger. That's my pinky. There's a complete map of the missing hand on the face. Now, why would this be? Here again is the medical mystery. I started thinking about Andrew inspiration from animal studies that have shown that if you cut the nerves going from the arms to the spinal cord, what happens is a complete reorganization of the sensory map in the brain. What happens in this patient when you remove the arm? You remember earlier when I spoke in the context of apotemnophilia, I said there's a complete map of the body surface on the surface of the brain, the postcentral gyrus. There's a vertical furrow on the side of the brain. Behind that is the map. Whatever. This map is systematic and point to point for the most part, but it turns out that the face area on the brain is right next to the hand area of the brain. It's a quirk and nobody knows why. The map is continuous and systematic, but oddly enough, the hand area is right next to the face area. In an adult, if you remove the arm, the hand area of the brain is now devoid of sensory input. It ain't getting no play, bro. It's hungry for new sensory input, and it's not getting any sensory input. The sensory input from the face skin, which normally only goes to the adjacent face area in the brain, now invades the vacated territory corresponding to the missing hand and activates the hand cells in the brain. That, of course, misinforms higher centers in the brain that the hand is being stimulated. The patient then experiences the sensations as arising from the missing phantom limb. When you touch the face skin, the message not only goes to the face area, but also activates the hand area in the brain. So you're getting cross wiring between the hand area and the face area of the brain. We did a 10-minute experiment to show this, and it challenged the doctrine in neurology that neural connections of the brain are laid down in the fetus and in early infancy, and once they've been laid down by the genome, there's nothing you can do to change these connections in the adult brain. That's why if you have a lesion in the adult brain, say following a stroke, there's such little recovery of function, and why neurological symptoms are so difficult to treat, notoriously difficult to treat. It was believed that there was no plasticity in the brain connections. We showed in our experiment that, in fact, there's a tremendous scope for rewiring. Excuse me. So much so that over a 2 centimeter distance in brain tissue in the cortex, the face input has now invaded the hand territory of the brain. Then we did brain imaging and showed that this invasion had actually occurred. But we already knew this from the psychological experiment. So I guess my mind is primed to think about cross connections in the brain. Now that's an example of cross connections caused by amputation depriving sensory input. In synesthesia, just like the face and hand areas, the color and number areas are right next to each other. I started thinking, well, maybe this is cross wiring again. But in this case, the cross wiring is not due to deafferentation. It's not differentiation. It's deafferentation. Because afferent are the signals going to the brain. Okay, and then so deafferentation is when you stop signals from going to the brain. So you've cut off this arm. Well, there are no more afferent signals from this arm to the brain. Okay, all right. Sorry, I just learned this. I didn't know that. But in this case, the cross-wiring is not due to deafferentation by removing the sensory input, but due to genes, given that it runs in families. Typically what happens is somebody phones me. It's usually a neurologist or a psychiatrist. They say, here's a strange case of a patient with apotemnophilia or Capgras syndrome or some such syndrome. Can you take a look at the patient and tell us what you think? The patient shows up in the laboratory or in my office and tells me what the problem is. I start thinking about it. You don't tell the patient ahead of time what your theory is because you don't want to cue them. Then you do various experiments on the patient and test your hypothesis about what's going on in the brain in terms of known anatomy and physiology of the brain, not some sort of mumbo jumbo psychological theory. Then you go and test the theory using brain imaging or doing simple psychological experiments. Sometimes we're able to devise treatment for the patients. For example, in phantom limbs, 2/3 of the patients with phantom limbs experience excruciating pain. There is no known treatment. I should restate that. There are 20 known treatments, none of them work. So we started investigating it to develop a treatment for it. But sometimes, even just explaining to the patient he's not crazy, telling him you've got a phantom limb, the reason for this is that something is going on in the brain, is a tremendous relief for him. Somebody has apotemnophilia and wants his arm removed, telling him you're not crazy, it's not Freudian, it's a specific anatomical reason why you're experiencing this. Then you go to the next step and say you have this hypothesis about what's going on. Once you've tested it, you know what's going on in his brain. But can you actually help the patient? In the case of phantom limbs, we've done experiments to show that we can. But let me give you another example. There's a curious disorder that I've not talked about in the past. Candy McCabe in England is studying it. And we are also studying it, and it's called RSD, or Reflex Sympathetic Dystrophy. Reflex Sympathetic Dystrophy. It's another one of these disorders that is not rare. I'd say 1 out of 20 stroke patients has it. You also see it in patients who don't have stroke, but have trivial injuries of the finger, like a metacarpal bone fracture that causes an injury with intense excruciating pain. It turns out there are two kinds of pain. We think of pain as one thing subjectively, but evolutionarily there are two kinds. There's acute pain, and there's chronic pain. Acute pain occurs when you touch a flame or a hot kettle and you say ouch, and you withdraw your hand. Chronic pain is when there's gangrene or a fracture, typically a fracture, and there's excruciating pain caused by the fracture and your hand becomes immobilized. You don't withdraw it. What's the evolution? Even though they feel the same perceptually, evolutionarily, they're very, very different. The function of acute pain is to mobilize the hand and remove it from the source of tissue injury to protect the hand. Chronic pain is the exact opposite. When there's an injury to a metacarpal bone, your hand freezes up and gets paralyzed temporarily. It's excruciatingly painful. Any attempt to move it is painful, so you don't move the arm. In the case of acute pain, you mobilize the arm rapidly. In the case of chronic pain, you immobilize it. Why? Because moving it could cause further tissue injury. So it's a protective reflex, immobilization. And then, of course, as the injury heals, you start moving your hand again and the pain goes away. That's a normal cause of events. But in a certain proportion of patients, stroke patients, and in a certain proportion of patients with a tiny little fracture, even a little hairline fracture or ruptured ligament, the pain persists with a vengeance. Even after the injury is healed, even as the fracture is healed, the pain persists for weeks, months, years, sometimes for life, for decades. Not only does the pain persist, the hand gets swollen and paralyzed. The pain spreads over the entire hand. This from just an injury to one little bone. And it involves the entire hand, the entire forearm. There's swelling of the hand, swelling of the arm, warmth, inflammation. All of that takes place on the arm. Again, you're stuck with it, and there's no known treatment that works. We started thinking about this and why this should be. Well, as I said, it's a reflex in mobilization, and it's painful. Anytime you attempt to move the hand, it causes excruciating pain. So the patient gives up and says, I'm not going to move my hand. Sometimes what happens is you get stuck with this, and this we call learned pain. Any attempt to move it, the signal that gets sent to the hand to move it, becomes associated with excruciating pain in your brain. So putting it crudely, you get a form of learned pain, a learned association between a motor command and the sensation of pain. The brain just gives up and the hand gets paralyzed. Any attempt to move it becomes excruciatingly painful. Excuse me. How do you break the cycle? We said, let's use a mirror. So we put a mirror in the center of the table. This is similar to a mirror treatment for phantom pain and for stroke that we discovered over a decade ago. You put a mirror in the center of the table, and the patient puts his painful, dystrophic, swollen, immobilized, paralyzed arm on the left side of the mirror. The shiny side of the mirror is on the right side, and the patient puts his right hand on the right side of the mirror, positions it so it mimics the posture and location of the hidden, dystrophic, painful left hand. He looks inside the mirror and sees the reflection of the normal hand. Suddenly, his hand looks normal, no longer swollen. That's obvious because he's looking at the reflection of the normal hand. And it looks like you resurrected his normal hand in the mirror, and it's optically superimposed in the position of the dystrophic, swollen hand. Now you ask him to send signals to both hands as if he were moving them, clenching and unclenching or rotating while he's looking in the mirror. Now he's going to get the impression, you don't initially ask him to actually move the left hand because if he moved it, it would be painful. He only moves his right hand and he imagines his left hand moving. What then happens is that the patient gets the visual image that his left hand, because he's looking at him, his left hand's actually hidden, right? So he's looking at a mirror, and in the mirror he's seeing the reflection of his moving right hand. So he's seeing his right hand move like that, and then he looks over here at the mirror, and he's still seeing his right hand move, right? Okay. What then happens is that the patient gets the visual image that his left hand, which is immobilized and paralyzed, is again obeying the brain's command. It looks like it's moving and is not painful. This way, you unlearn the learned pain and the learned paralysis. The astonishing thing is that the hand actually does start moving for the first time in his life. First time in decades, first time in years. It works better if you do it very soon after the dystrophy sets in, a few weeks or months afterward. The hand starts moving again and the pain subsides, and in a remarkable example of mind-body interaction, the swelling also subsides, often in a matter of hours. This chronic pain disorder is considered intractable, incurable. It has been known for decades. I think it was discovered more than 100 years ago, for which people have done dorsal rhizotomy, cut the nerves going to the spinal cord, cut the spinal cord to treat it. They do a sympathetic ganglionectomy that does work to some limited extent. You can treat it equally effectively, if not more effectively, with just a $2 mirror. The patient looks inside and moves his normal hand. We suggested this therapy some years ago, but it was actually Candy McCabe who first properly described it. We suggested the idea, but she discovered it independently. There have been clinical trials on this from a group in Germany, I believe, on 50 patients. The discovery was originally made on a handful of patients. Since then, there have been double-blind, placebo-controlled crossover trials, which is the best type of clinical trial you can do. And people have found dramatic recovery from this pain in a matter of a few weeks of mirror treatment. Then the pain stays gone for a period of at least 6 months, and then you may need a refill after that. Imagine the amount of pain and agony and invasive surgery this has saved. Sometimes you come up with an off-the-wall, half-plausible hypothesis, and there can actually be a clinical use for it. One example is RSD, or reflex sympathetic dystrophy, now called complex regional pain syndrome. We've talked about synesthesia. We've talked about apotemnophilia, and we've talked about reflex sympathetic dystrophy. There are other syndromes like this that we've studied. Another syndrome is called Capgras syndrome. This one's my favorite, by the way, and this is me speaking. That's the Capgras. I learned it from Ramachandran, and I think it's pretty cool. There are some syndromes like this. There are other syndromes like this that we've studied. Another syndrome is called Capgras syndrome, where a patient has been in a coma for a week or two and comes out of the coma. He's a little bit slowed down. He has mild dysarthria and problems talking, but is otherwise mentally perfectly lucid and normal and can hold a fluent conversation, can play chess with you, can do arithmetic. Everything seems fine, except a little bit of slurring of speech. This chap looks at people and can recognize them no problem. He's not psychotic, not mentally disturbed. The conversation is normal, except for the little bit of slurring. He looks at his mother and says, doctor, this woman, she looks just like my mother, but she's not. She's a stranger, some other woman who looks like my mother, but in fact is not my mother. She's an imposter. Sometimes it develops a paranoid touch. He says, why is this woman following me all the time? She's not my mother. She's pretending to be my mother. Why does this happen? The Freudian explanation again. By the way, I don't mean to do too much Freud bashing. I know it's fashionable in New York, but I think that he had deep insight into the human condition. Oh, what's fashionable is bashing, Freud bashing. But I think that he had deep insight into the human condition, especially the role of the unconscious, which we are increasingly realizing is largely true, and Eric Kandel has written about this. But anyway, it's fun to do so. So some Freudians had a theory about Capgras syndrome, that when this chap was a young baby, when he was an infant, he had a strong sexual attraction to his mother. Freud called it the Oedipus complex. As he grew up, the cortex developed and started inhibiting these latent sexual urges toward his mother, and therefore, as an adult, he's no longer sexually turned on by his mother. But then a blow to the skull damages the cortex, and these flaming sexual urges come to the surface of consciousness, and suddenly and inexplicably, he finds himself sexually aroused by his mother. He says, my God, this is my mom! How can I be sexually turned on? This must be some other strange woman. This is, of course, a very ingenious idea, as indeed a lot of Freudian ideas are. It doesn't work because I've seen patients, at least one patient, who had the same delusion about his pet dog, pet poodle, saying, Doctor, this is not Fifi. It's some other dog pretending to be Fifi. Now, if you try to apply the Freudian analysis to this, you've got to start talking about the latent bestiality in all humans and some rubbish like that. So it doesn't work. This got me thinking that there's something going on that's probably a neurological in the brain. I'm mainly an experimental scientist, and we go with the flow. It's like charting the source of the Nile. You don't know when the next surprising twist and turn is going to be. It's a great adventure, a grand love affair with nature, with all these twists and turns and unpredictable events. Oh, excuse me, that's how we do experiments. But all of it is headed toward the goal of understanding human nature, but understanding it piecemeal. For example, you can't ask, what is consciousness? Some people do, but it's too nebulous an idea. In fact, philosophers have criticized this approach. But I think it's okay to ask questions like Francis Crick did. Well, what is consciousness? Philosophers like Colin McGinn and others have argued that this is utterly mysterious. The human brain can never comprehend itself and can certainly not comprehend mysterious phenomena like consciousness. Somebody like Crick would vehemently disagree, and I would agree with Crick. Crick and Koch, for example, have argued that there is a structure called the claustrum that is a thin layer of tissue underlying the insular cortex of the brain. What's exciting about this layer of tissue, what caught Crick's eye and Koch's eye, was that it doesn't have any known function, unlike other regions of the brain. There are many regions that we don't know the function of, but the claustrum is especially mysterious. It's not a tiny little structure, it's a medium-sized structure, and it's homogeneous in its cell constituents. It also doesn't have the layered structure as with the rest of the cortex. The astonishing thing Crick noticed was that it's connected to almost every part of the brain, including every part of the cortex. And it seems reciprocal. It sends connections to the somatic sensory cortex and receives connections back from the somatic sensory cortex. It sends signals to the amygdala, back from the amygdala to the anterior cingulate, back from the anterior cingulate. In fact, it's very hard to find any region of the brain that is not connected to the claustrum. John Smythes in our lab, and I have now picked up the gauntlet where he left it. Crick, for example, has been rewarded in the past for analogies, for big metaphorical leaps. I don't think he actually says this, but if you look at the double helix and the complementarity of the helix, the two sides of the helix, we're struck by the analogy between this and the complementarity between parent and offspring. There's a huge leap of faith there. He says, why do dogs give birth to dogs and not to pigs? Any child will ask this question. You and I won't. But Crick asked that question. Why do dogs give birth to dogs and not to pigs? There's a complementarity between offspring and parent. Might it be the case that the complementarity of the two strands of the helix actually dictates complementarity of offspring and parents? This was the big leap. Then, of course, he figured out the genetic code and modern biology was born. He's primed to think in terms of linking seemingly unrelated phenomena, of linking structure and function. Then he approaches the claustrum and says, what's the most fundamental thing about consciousness? So axiomatic, in fact, that you take it for granted? That is the fact that you are one person. Right? That's the illusion, right? That you are one person, unity of many attributes of human consciousness, the continuity, the time travel, the ability to go to and from in time, looking into the future, visiting nostalgic memories from the past, stringing them together in approximately the right sequence. Laughter is uniquely human, and we can't imagine laughing without being conscious. Many attributes of human conscious experience, self awareness is another attribute. Putting it crudely, consciousness is aware of itself. Now, the central attribute of human conscious experience, so fundamental in fact that we take it for granted, don't pause to think about it, is the sense of unity. You've got a diversity of sensory experiences. You see things, you listen to things. This harks back to what I was saying about synesthesia. You taste things, you have hundreds of memories throughout a lifetime, yet you think of yourself as a unified person. Yet all of these happen to you. You, John, or me, Rama. It all happened to me, and I am one person. Despite this diversity of sensory experiences, this bewildering sensory cognitive blitz of memories and sensory impressions, I experience unity. How does that come about? Another way to formulate this question is that there are different brain regions actively processing different aspects of information, including memories, and yet you experience yourself as a unity. Many philosophers will argue that this is a pseudo problem, not a true problem. In fact, Crick adopts the opposite view. He and Koch debunk the idea that it's a pseudo problem. He says that the most axiomatic thing about consciousness is its unity. And guess what the claustrum is doing? It's getting sensory inputs, even inputs from the motor cortex. It's getting inputs from every region of the brain in one little medium size gathering place and sending messages back. It's ideally suited for performing this unifying role. There's an analogy here between what the structure of the claustrum is and what the phenomenology of consciousness is. Maybe this is not just a superficial analogy. Maybe it's deep. Maybe the clue to consciousness lies in looking at the structure of the claustrum, a detailed study of its microanatomy and its connections to the rest of the brain. Questions of that nature, trying to explain functions like consciousness, like self-awareness, like qualia in terms of brain structures, is something that Crick pursued, and I think it's something that I'd like to pursue as well. And we have been trying. We all share his agenda, though obviously not his stature. That could have been edited down, bro. That's all right. All right, done. Oh. That's not too bad, bro. All right. Oh, bit late for a walk. That's all right. Sorry. Okay. Good night. Bye.